Welcome back. And I was telling you about the different kinds of polymerase enzymes which are obtained from different sources. We require two things essentially. One is that the polymerase enzyme should be heat stable so that we do not have to add the enzyme again and again for every cycle. Okay. And the other important feature of polymerase enzymes is that they should be high fidelity enzymes so that they will be able to copy the template DNA exactly as it is, and there will be minimal chances of any error in the final product of the DNA. Okay, so we have seen that Taq DNA polymerase is one such enzyme, but the problem is it is not very faithful. It is not a high fidelity enzyme, and therefore other enzymes are also used. For example, the Thermus brochianus organism is used for isolating polymerase enzyme, which is called as TBR enzyme, and there is another enzyme called T4 polymerase, which is taken from E. coli cells, which are lysogenized with T4 phage. Okay. Do you know what is the meaning of lysogenized? E. coli lysogenized
Yes, sir.
phage. Okay, it means that the DNA of the phage is integrated with the host DNA, with E. coli DNA, and therefore, whenever this E. coli DNA replicates, the phage DNA will also replicate. That is one thing. And the other thing is that whatever genes are there in the phage DNA, these genes will be expressed by the host system. Okay, so in this particular case, the T4 gene is there, the polymerase gene is there, and that gene is expressed by the E. coli cells, which are lysogenized by the phage. Okay, so long as the phage doesn't go into the lytic cycle, the host is safe, the host will continue to survive. But once the phage takes the decision to go to the lytic cycle, then probably the cell will be at a risk, the cell will be destroyed because the phage will multiply inside the cell, and when a large number of phage particles are produced, they will lyse the cell and come out of the cell. Is it okay? You might have already studied about
Yes, sir.
phage genetics in your TYBSc. Okay. Then, so what we therefore need is an enzyme which can tolerate high temperature, and another enzyme which can copy the DNA very properly, and therefore usually combinations of different polymerases are used. For example, I have written here Dynazyme. Okay, this is actually a brand name for an enzyme which is produced by an industry, by a company. Okay, so this Dynazyme actually is a combination of two enzymes. One is the TBR, which is a high fidelity enzyme, and the other is Taq polymerase, which is a high temperature resistant enzyme. Okay, or thermostable enzyme, right? Then there is another product which is called TAQ, TAC plus. Okay, and this is particularly used for long PCR systems. What is a long PCR? It is a PCR reaction in which the length of DNA, the template DNA, is very large. Okay, long DNA strands can be copied, and for that we have a combination of TAC polymerase, and the other polymerase is called PFU. Okay, and this particular enzyme is taken from another thermophilic organism which is called as Pyrococcus furiosus. Okay, Pyrococcus furiosus, PFU. Then the next point is about the designing of primer. This is a very important topic, and the reason is that if the primer is not correct, if the primer either if the primer is not correct, or even if the primer is correct but it doesn't bind to the proper site on the template DNA, then the product which will be formed, okay, the newly synthesized DNA will not be the copy of the original template DNA. It will be something else, okay, which we don't want to happen, and therefore designing the proper is a very important part of the PCR reaction. Okay, essentially the primer must be complementary to the sequences which are flanking the target DNA. What is the meaning of this? Sequences flanking the target DNA. Okay, I go back to my diagram here. Okay, so suppose this is the region which I want to copy. Okay, this is the target DNA. Okay, then this particular part of the DNA, which is a sequence recognized by the primer. Okay, primer is binding here, primer is binding here. Okay, so this particular sequence is very important, and the primer must be complementary. It must be exactly complementary to the sequence which is at the extreme part of the DNA that we want to amplify. Okay, and this particular portion is called as the flanking region. Okay, the region which is flanking the gene of interest, okay, or sequence of interest here, that sequence is very important. The primer should actually recognize and bind to this particular sequence. Is it okay? Any question? Then another thing is
No, sir.
this particular primer must not amplify any unwanted sequence. It should not go and bind to any other region on the DNA. Okay, and therefore it also means that whatever sequence is there at the extreme end of the DNA or the flanking region, that sequence should not be present anywhere in the target DNA. Okay, if the same kind of sequence is there anywhere in the DNA, then the primer might bind to that particular portion, and therefore the product which will be amplified will not be the desired product. Right? Then another thing is the primers must not be self-complementary. We are going to add two primers, isn't it? One of the primer will bind one strand of the DNA, and the other primer will bind the other strand of DNA. Now these two primers should not be complementary to each other, because if they are complementary to each other, then they will bind to themselves. Okay, one primer will bind the other primer, and they will forget binding the DNA, and therefore the particular product of interest will not be amplified. Okay, target DNA will not be amplified. Is it okay? Then another thing is the GC of the primer should be roughly similar to the GC content of the target sequence, target DNA. Okay, so that is the GC percent, we call it. Suppose it is 20%, then if the primers contain 20%, if the target DNA has 20% GC content, then the primer should also contain 20%. Why that is essential? Because in the melting or denaturation step, okay, at 94 degree, when we want the two strands to separate from each other, okay, if the primer GC content is different, then there is a probability that this primer will not get separated from the DNA easily. Okay, although the other part of the DNA will get separated, the primer part will not get separated easily. We don't want that to happen. Okay, the entire stretch of DNA, that is the two strands of the DNA, should get separated from each other at the same time for all the molecules of DNA. Is it right? And therefore the GC content of the primer should be similar to the GC content of the target DNA. Okay, then sometimes what happens is, if it is for example a quantitative PCR. Quantitative PCR means what? While the DNA is amplified in the PCR reaction, at the same time the machine will automatically count the number of DNA molecules, or it will give you the it will tell you about the amount of DNA that is synthesized, okay, at every cycle, in fact. Right? So for this kind of detection, there are fluorophores, or fluorescent compounds are bound to the DNA molecules. Right? Now the fluorophores are actually bound to the primer DNA. Right? So these primers, if they have the bound fluorophores, then it is easy for the quantitative PCR work. Usually in a reaction mixture, we add about 0.1 to 0.5 micromole of the primer, each primer. Okay, and that is to say we are actually adding about 6 into 10 raised to 12 to about 3 into 10 raised to 13 molecules of the primer. Okay, and then we should know that this particular supply of the primers should be non-limiting. Non-limiting means what? It should not be the rate limiting amount. If it is rate limiting, that what will happen? The primer molecules will get exhausted at a particular cycle, and there will be, if the number of molecules which are already synthesized, if it is more than the total number of the primers, then what will happen? Further amplification of the DNA will not take place. Okay, only a fixed quantity of DNA replication will take place, but most of the DNA molecules will be there which will not have the primer molecule and therefore no amplification for those particular strands of DNA. Amplification will not take place. Are you getting what I'm saying?
Yes, sir.
Okay, then I think the time is already getting over, so I have to stop this lecture now. The remaining part about this primer, I'll discuss it in the next lecture, because it will take some time for me to explain all that. Okay. So in the meanwhile, if you have any difficulties, any queries, you can ask me. It's about 12:23. So at 12,
No, sir.
yeah, 12:30, madam will start her lecture. So at least 5 minutes I have to give you in between. Okay. Yes, any question? Amol,
No, sir.
who was
No, sir.
Amol? Inamul, any question?
No, sir.
Nurada,
No, sir.
Shamata,
No, sir.
Shraddha,
No, sir.
who else is there? Bharat Wabre, Yogita Desh,
No, sir.
Yogita, Minal, Pooja.
No, sir.
So if you have no question, then I will end this meeting for today. Tomorrow we'll again meet and try to complete the most of the other part which is remaining. Okay, and then maybe in the third lecture from now, I will tell you about the PCR machine. Okay.